No headset yet. I gotcha. Turn me down, though. 222. Check, check, check. Hello, hello. What was that? No, it was a frame. We just lost video on Herc there for a second.
Okay, I'm on SPL here. We just changed our watch over, and yeah, we're ready to get started here with any and all science that the back row has to direct.
Thanks, Steve.
It was Rennie, but I sound like Steve.
Thanks, Rennie. Actually, at this time, if we're all settled, we're going to be going into radio silence to conduct a private ceremonial protocol and acknowledge and honor the biocultural resources that are present and that we're interacting with and impacting. So we're going to do this once at the beginning of the dive to really set our intention for our watch here. And we'll be back on SPL shortly. So I'm going to invite everyone to go turn your mic off, and we're going to together collectively enter this time together. Mahalo. Invite everyone to turn your microphones back on. If anyone out there listening has questions or is interested in the work that we're doing alongside the Hawaiian Cultural Office of Hawaiian Cultural Affairs and our Hawaiian liaisons aboard ship, I invite you to read some of our recent blog posts on nautiluslive.org.
Aloha front row, this is Beth.
Hey, Beth.
Hello. Before we start moving, can I ask video to zoom in on what we're looking at just so I can get a better sense of the texture of these rocks, please?
Sure, stand by here.
That's great, thank you. Yeah, these don't look like they have very thick crusts on them. That's okay. Just trying to get a better sense for what I'm looking at here so that I know when to see something that might be different for collecting for our purposes.
What are you seeing that tells you it's a thick crust, Beth?
Yeah, so these basalts that were formed on these seamounts, kind of like lava on a volcano, they get, actually what the lasers are looking at right now is maybe a good indication where you get these kind of, I describe it as like an Oreo cookie crumble, like if you take the darker parts of an Oreo cookie and imagine squishing them up and kind of glomming them onto a rock. That's an indication of manganese oxides, which are the alteration products that form. And so those are kind of the textural cues that I'm looking for, that there might be good crust on the rocks.
Jeff, you want to come wide for a minute, please? Thank you. Let me get you a better shot. Sorry. Drifted off my set.
Feeling some current down there?
A little bit, but not much.
And can I ask the front row, do we have a ships move in already, or can we spend a little bit of time here picking things up?
We are stationary right now.
Okay, great. Yeah, so I might like to try to pick up a couple things and just give them a little squeeze with the manipulator.
Sure thing.
Where we just were, off to the upper left,
Sure.
looked like it might have some material that might be of value. Yeah, we'll get out this little doohickey here in the back row. Yeah, so in this area, looked like there were some darker pieces.
Roger.
And I'm going to change this. There we go.
And for those of you who weren't able to see what Beth just did, she did a magical touchscreen arrow using the Telecaster, which is really cool.
Yes.
Does that show up?
I don't think it shows up.
Okay.
Give me a second, we'll try and put it up.
Someone may correct me.
We seem pretty stable here.
Yeah. So Beth, anywhere dead ahead type?
I'm sorry, was that a question to me?
Yeah, is dead ahead, is that the area you were eyeing before?
Yeah, so some of these we might not be able to pick them up. Can we zoom in on this area here,
Sure thing.
the video?
Coming in.
You can push it in
Yeah,
a
so
little
I
bit.
might like to try like this piece right here, maybe? And some of these other ones that look kind of crusty. See if they're not actually attached, and we can maybe try to pick them up.
Sure thing. All right, go ahead and come full wide, please, Jeff. A little too far there.
Yeah, these in particular look like they might be promising for the crust. It's just a matter of whether we can actually grab them.
Nice Oreo cookie rocks.
Yeah.
Well, if you're wondering who the new voices are, feel free to check nautiluslive.org homepage. You can see the 8 to 12 shift. We'll do introductions a bit later. All right, Jeff, you want to go in and push it on that rock, please? It might be pretty attached, this one, Beth, but we can give it a go.
What are we looking at? Yeah. So maybe this feature right down here might be
Okay, the bottom corner there.
not, maybe we're lucky.
Roger. All right. Go ahead and come full wide, please, Jeff. And did you want scaling lasers on this before or after we attempt?
Right now is fine, as long as I'm assuming there's some automatic pictures being taken.
Roger.
You can
Yeah,
take
that's me. I'm taking images
Yeah.
for the sampling.
Yeah, so I think we're good. And if that piece there is not good, maybe one of these over here is also a potential.
Roger that. And go for a poke first, is that the... Did you want them to poke it, Beth?
I'm sorry, I didn't hear the question. I need to turn my volume up.
Go ahead and push on in there, please, Jeff. That's great. So Beth, you want us to give it a poke first?
Yeah, that sounds great.
Great. There's a little bit of
Yeah,
material
so
here.
a little bit friable.
Otherwise.
Yeah, so that's promising.
Yeah, that piece in there is definitely moving.
So Beth,
Yeah,
what would you like us to do? Would you like us to take that sample?
if you think that you can grab onto it and yeah, kind of wiggle it loose, I think it's worth giving a try.
Okay.
This keystone's going to take the whole C-mount down.
Maybe I'll change my grip force in case it's a little bit friable.
Could we get a zoom in on that before we try?
Sure thing.
Is that possible?
Yeah,
Go ahead, Jeff.
Yeah, thank you.
Good. Got some images of that. Is that full zoom?
Roger. All right, Jeff, go ahead and come a little bit wide, please. A little more would be great. Thanks.
Looks like there's a piece up at the top there that just wants to come right off.
Yeah, that one's cooperating with us. Is that yellowish color part of the rock, Beth?
It's a little hard to tell from this viewpoint, but it looks to me like it might be like a clay alteration product that often forms on the inside of these manganese crusts. If you remember some of the images I showed the other day, when you break open these materials, often you'll see clay or even sediment trapped on the inside.
Jeff, go ahead and fish on in there a bit, please. Interesting.
That piece that was a little bit above the manipulator that was already kind of coming off, how
Looked like
big
it
do we
dislodged.
estimate that at? That's probably like 5 or so centimeters.
You want that piece instead?
Maybe we can just pick it up and see,
Roger that.
have a look at it.
Go ahead and come a little wider, please. All right, go ahead and push them back in there, but thanks. A little wide there, please, Jeff. That's great.
Looks quite friable.
Yeah.
Oh, it just rolled over.
I wonder if this piece comes off, actually.
The big one.
Bigger one, yeah.
It's worth a try if you think you can get it.
Oh yeah, that's way better.
Quite friable. Oh no, it's still there. That's all right.
Yeah, so that's a nice small piece. Can we zoom in on that piece a little bit?
For sure.
Oh yeah, that's a nice altered glass, it looks like. Oh yeah, and then that's definitely altered. Okay. If we could put this in one of the forward bio boxes, we might try to collect another small piece and put it in the same spot, but we might as well go ahead and keep this one while we have it in the manipulator.
Sure thing. All right, go ahead and come forward, please, Jeff.
And this would be all part of the same sample?
Yeah,
Yes, okay.
exactly. I would treat it as the same sample.
Yeah, yeah.
Science, is there any floaty samples we need to know about in the front?
There's nothing in the front.
Nothing in the front, Raj. Jake, you want to go ahead and pop out that toolbox there?
Popping out.
What is this sample number, Steve?
004.
Thank you.
So NA134-004.
Thank you, Jake. If you wouldn't mind following me in on the bubble, please.
Yep.
That's going in compartment B for
Okay.
back row reference. So the right side we call B, and the left side is A.
Well, I like it going in the B box first because this is the Beth box.
All right, Jake, go ahead and close that up, please.
Yeah, a lot of space in that box. Yeah, so if we could try to see if there's any other pieces in that same feature that we were just poking at.
Sure thing. Go ahead and push on in there a bit, please, Jeff. That's wonderful. Yeah, so there's that small piece that rolled down the hill. There's also this larger piece in the back here.
Yeah, if we could try for the larger piece, that might be better material for the kinds of analyses we'd like to do.
Sure thing. Actually, let's try this.
For those watching at home, you might be wondering why I'm interested in a rock like this. So
Ooh, that big piece.
That big piece might have moved.
The research that my team is interested in exploring with the samples that we collect on these seamounts is to understand both the mineral content that forms in the crust on these very old rocks in terms of the types of metals and minerals that may be in them, as well as the microbial communities that are living within these crusts and maybe performing functions like supporting primary production, which is the fixation of carbon, just like plants do. But instead of using sunlight to fuel that, they're using chemical energy. So we're trying to understand the types of microbes, what they're doing. Oh, that looks beautiful.
Nice.
Excellent
Excellent.
work.
Yeah.
Thanks, Jess. Excavation mode.
You have a bright future in dentistry.
So if we could hold that up again to get a nice glamour shot before we put it in the box, I'd appreciate it.
Sure thing. Jeff, you want to come a little wide there, please? That's wonderful. Thank you.
And if folks were around earlier and saw our previous rock sample, it was quite different than this. Why are we looking at such different types of rocks?
Yeah, thanks for that question, Lindsay.
And maybe if you can't answer it now, that's fine.
So there are different objectives that we would like to apply to the different rock samples that we collect. As I was just describing, my work is interested in the alteration of the rocks.
All right, go ahead and push on in there a bit there, please.
Lots of different coloration.
Yeah, so you see some iron oxide staining potentially there. The black is probably some manganese oxide that also co-precipitate many other types of metals. Doesn't seem that we have ground mass in this piece, but that's okay.
I was going to ask, is there any of that original?
Yeah, so this piece won't be great, most likely, for the dating aspects. But that, I think we're going to have a hard time finding pieces that are great for both of our types of analyses.
Sure.
Thank you so much for that glamour shot, and we can go ahead and put that in the same bio box.
Sure thing. Full wide, please.
All
right, Jake, and when you're ready.
So the other rocks
go ahead
that were picked up earlier in the dive were specifically targeting rocks that didn't look like they had been highly altered and were more of the original material, the original composition of the rocks. And that's because we want to be, the scientific team wants to be able to get at certain mineral grains that would enable them to date the age of the rocks. And pieces like this will be too highly altered the signal will have been lost of the age.
So the 8 to 12 watch, we have
Jake,
Dr. Beth Orcutt here, who is interested in those altered rocks. If you do stick all the way around until the 12 to 4 watch, you'll get to hear our scientist, Andrea, on board, who will be interested in those harder rocks in order for dating.
Beth, you want any more rock samples around here, or are you good?
There was that other small piece that had already dislodged. Couldn't hurt to put that in the same spot, and that might be a good specimen that we don't subsample.
Sure thing.
That could be used as an archive piece, potentially.
All right, go ahead and push on in there a bit there, please. Also floating off my spot. The one kind of center screen now. You want to come a little wide there, Jeff? That's great. This guy here?
Yes.
Great. Rising land into crowds.
Yep.
And come full wide there, please. Sorry, I just pushed myself off my parking spot.
If it's too difficult to reach it at this point, that's okay too.
Oh no, all good. It'll take one moment, sorry.
Steve, what is our current depth here?
Depth?
Yeah. Oh wait, it's right in front of me.
Oh yeah,
Sorry.
if it's blocked, you can just move the window to the side. It might be behind that window.
Thank you.
There. 2563.
All right, so we still got a ways to go up this mountain.
All right, Jeff, go ahead, push on in.
Indeed, we have traveled about 300 meters, and looks like there's about 1,000 meters to go from the original end target. In the XY direction, not depth.
All right, go ahead and push on in there, please.
A small but mighty piece. Okay, thank you.
All right, full wide.
Thinking the current must be
Jake.
strong or something, because
I'm going to go ahead and pop her out.
Yep. Coming out.
Roger. Are you ready? Oh, yeah.
I've been watching it.
Oh.
So much alteration, it doesn't want to let go.
Oh, interesting.
Is that because it's almost spongy?
Well, the mineral alteration products like clays and things, imagine, you know, clays are very sticky.
All right, Jake,
So-
go ahead.
Sure.
Okay.
Jess, that was impressive. Do you have any kind of feedback on the mechanism to tell you what kind of pressure to give?
Nope, just visual. There is a force feedback option, but we don't use it.
Okay. Thank you so much for that sampling, and thank you to this spot for providing a rock for us for our analyses. I believe we'll go ahead and set in a ship move to start moving back up this ridge.
Okay.
And scanning around to see what kind of animals we see.
Looks like if we go due south, I don't know if you have the HIPAC survey, HIPAC 2 screen up. Argus has drifted a little bit off to the east, but I think if we just go due south, we'll eventually rejoin our ridge here and get some of this steeper slope on the way. Is that good for you, Beth?
Yes.
Okay. Let's go with, let's do 20 or 30, tether length, 30 meter.
Yeah, 30
stuff.
meters.
Bridge nav. Yes, can we have a ship move 30 meters bearing 180? Thank you.
So for those who might not have seen the earlier shift, there were some interesting overhangs as we were moving up the ridge. So definitely some dynamic features on this seamount flank.
That's right, and some of those features provided really good habitat for a number of different organisms, and sponge and coral, invertebrates.
Yeah, so we'll definitely want to keep an eye on the Argus cameras, which give us a great view of what Hercules is about to see.
Yeah, that Argus camera can be seen on your quad view. I believe it's the upper right. Is that Jeff? Is that right, Jeff?
Yeah, it's number 2, the upper right.
So you can see that Argus is keeping an eye on Hercules. You can see that slightly wider view. Sometimes you see things ahead of Hercules. And since we have switched out, I wonder if we can go around the room, do a quick introduction. Would now be an okay time?
It's a great time.
Excellent. I'll go ahead and start off. You can also see our names and click on our bios, learn all about us from the Nautilus Live main website here. But my name is Lindsay Holliday Van Dam. I'm the Science Communication Fellow for this 8 to 12 shift. And for those of you asking, we are in Hawaii Standard Time. I'm going to pass it off to my right.
Thanks, Lindsay. Hi. I'm Beth Orcutt. I'm a senior research scientist at the Bigelow Laboratory for Ocean Sciences, where I study microbes that live in the deep ocean, try to understand the roles that they play. And this is my first cruise, really, with the Nautilus. So I'm really excited. My first dive as well. Thank you for having me.
Aloha pumihana kākou, mai ka mokupuni o Hawaii o ke awe ahi ki loa aku I holani kui papahana o moku a ke nei. O no ke e o kalama ehu Takahashi, o me ina no ka waha olelo ma ka olelo Hawaii ma ke iuhu ka i o kākou ma ka moku nui o Nautilus nei. No leila, e nā Hawaii ke olu mai, e hea wale mai, nau iaku ke la mau ka hua punai vere a waiho ko mau mana o mana olelo e lua. Lea ka wale mai, e imi wale mai I manawa e hui ai me mākou ma Nautilus.org. Aloha everybody, my name is Kalama Ehu. My role here on the Nautilus for this trip is the Olelo Hawaii Liaison, or Hawaiian Language Liaison. We'll be providing any sort of commentary that we can, but we please urge you guys, all of our Hawaiian language speakers out there and communities from Hawaii to please check us out at nautiluslive.org and please ask us questions in both languages, both Hawaiian and English. We'll be on from 8 to 12, like you said. And yeah, oihola, mahalo.
Hello, everyone. My name is Steve Voskovich. I'm sitting in the data logger chair. I'm the science manager aboard Nautilus for this cruise. I am also a visiting researcher at Boston University, where I study deepwater coral biodiversity. Front row.
Yeah, this is Renato Kane, the navigator on this watch. I've been with Nautilus for quite some time, and I'm happy to be here and just outside of Papahanaumokuakea Marine National Monument, soon to be inside.
And we've heard Jeff's voice, but do you want to introduce yourself, Jess?
Oh.
Just kidding. That was only for the front row to hear. They already know who I am. Hi, my name is Jess Sandoval. I'm sitting Herc for this 8 to 12 session, and Yeah, excited to be here, as per usual.
And I'm Jake Bonney. I'm sitting in the Argus seat. And I just finished up my master's in ocean engineering at the University of Rhode Island.
Woohoo! Yay.
And I'll round it out with I'm Jeff Dennerline. I'm the lead video for this particular cruise. And as usual, it's always fun to be out here.
Thanks, everyone. As you can see, it is a rock star watch here, so stick with us.
Good pun. We love rocks.
Starting to see a little movement in Argus there.
Yeah, roger that. It's a beautiful structure. It looked almost like an octopus. Definitely
yeah, like a little cradle.
Yeah, some nice broken pillow basalts here.
Yeah.
Beth, do you need water collections paired with your samples for filtering?
Thank you for bringing that up, Steve. That would be great.
Okay, we can stop anywhere and do a Niskin bottle. We have 6 available, each of 5 liters.
Yes.
So we can pop as many as is needed.
I only need 1 for my work, so we'll save the other ones for Meredith's eDNA. And this sample won't be great for that because there's not a lot of animals. So yeah, front row, if we could fire 1 of the Niskin bottles.
Sure thing.
As a reference for my sample.
Sure.
Is there a preference on altitude?
I don't have a preference on which number.
On altitude.
Oh, what are we at right now?
We're at an altitude of 4.6 meters.
That's fine.
Roger. Okay. I'll just bogey a little bit forward and then we'll take one. All right. And there have been no, actually I might just move a little forward here. There haven't been any Niskins that have been sampled currently?
None.
Correct.
Okay, great.
And for any, for video, do we have a way to see the outdoors to see if the eclipse is happening?
Oh.
Yeah, we might. Let's see if I can find something here.
That was the alarm that just went off. That's my reminder that the eclipse is
I
happening.
was wondering what that was.
And just for a quick translation, if anybody was wondering out there, so the Hawaiian word for the eclipse of the Mahina is going to be Poli Kamahina. Poli Kamahina.
Oli
Basically means that, yeah,
Right.
Poli Kamahina, which is basically going to mean that the moon is darkened.
Okay. Thank you.
Well, unfortunately, there's a portion of Nautilus that's kind of right in the way.
All right. Looks like we're going to hit up number 6, I suppose. You guys got eyes on the Niskins there?
Yep, watching.
Roger that.
So for those watching at home, not knowing what Niskin means, Niskin is a name of a type of bottle. And what we're doing here is pulling a trigger to close that bottle so that we capture a water sample, roughly about 5 liters. And I'll be using that to understand the microbes living in the water around the rocks that we collected a few moments ago.
All right, ready, set, fire.
There it
Up.
goes.
There it goes.
Kind of cool how that mechanism works. Just that.
I was taking at 4 meters of altitude in case you guys need that for your...
Quick tug. And
Sample 005, correct?
That's 5, yeah, sample 5.
Thank you for that reminder, Steve.
Argus has just about completed that move. I'm seeing, yeah, roughly a delay of like a minute per 1,000 meters from when we call it in to when it happens, and similar on the tail end.
Okay.
Well, Lindsay, unfortunately, there is a rail.
Of course
on
there
the
is.
port side that is doing a nice job of obscuring our view of the lunar eclipse. Which is, actually our dynamic positioning is doing great because the pole hasn't moved, so.
Good to know. Yeah, I'm still seeing quite a bit of that eastward
Sure thing.
current or whatever that's affecting Argus. You want to call in, would you like me to call in another move and keep moving along there, Beth?
Yeah, if you think we need one to keep moving.
Yeah,
that makes sense.
We're just about stopped, so.
Okay.
We have some room to explore while we do bridge nav.
So on channel 3, set channel 3, which is the bottom left of your
One more
quad,
step, 30 meters, bearing south.
We see the eclipse right now.
Thank you.
Can you tell us a little bit about most of what we're seeing here? If most of the rocks, are we looking at some of that altered rock, or are we mostly looking at older, unaltered?
So, yeah,
if you can say.
Yeah, sometimes it's hard to say from a zoomed out view like this. So we're definitely looking at seafloor basalts, and you can tell by the structure of the rocks that the conditions of the lava that formed it were slightly different. So sometimes you see kind of what look like pillows, big blobs, right? So, and other times you see what looks more like a flow. And so the composition of the molten rock is different in forming those. And so that's why they end up having different structures.
Is that based on factors like temperature and current?
No, not necessarily bottom sea conditions, but more the conditions of the melt itself, and things that are dissolved in the melt that give it more or less viscosity, more or less porosity. And yeah, the conditions that the molten rock is moving through.
Thank you.
And so here we're seeing some pillow type features, some of which are broken. And then occasionally we're seeing a couple little attached creatures. Nothing quite huge and dynamic yet in our field of view. It looks like it's going to get a bit steeper ahead of us.
Yeah, so for those of us just joining us, we are inside Papahanaumokuakea Marine National Monument. This is our first dive of this expedition, the Lu'ua Ia Ahiki Ika Papaku. And we're really excited to be here. We've launched the ROV at our deepest depth, about 2,700 meters, and we're slowly making our way up to where we anticipate leaving at around 2,200 meters deep.
Just a technical clarification. I believe this seamount is just outside the boundary of the monument, but we will be entering the monument shortly.
As part of this dive?
Yeah, that's a good question. I'm not sure if the entire seamount feature is outside of the monument boundary.
Yes, it is. This seamount is entirely out.
Yeah. So our the remaining dives of our expedition will be within the monument.
But it's just outside.
Yeah, and we decided part of the motivation to come to this seamount, even though it's outside of the monument boundary, is because we want to understand the ages of the seamounts in this feature that go into the monument almost as a chain. And so we want to start at one end where they might be younger, and then move towards the northwest end where the seamounts might be slightly older.
Current's picked up a bit over here.
Yeah, it's kind of seems like it's really swirling around this area.
Yeah.
We're really fortunate to have Kalamehu Takahashi here on our watch. I invite you to, at any time, please.
No, yeah, and then mahalo for the introduction. Just to provide a little bit of context into the name that was chosen for the voyage. So at this time, we'd like to definitely send out our acknowledgements to the Cultural Working Group, which is like basically the Cultural Advisory Board for NOAA. And so in the process of deciding the name for these voyages, we decided to stick to some very like practical and like utilitarian terms in order to describe maybe outside some of the deeper cultural meanings, but more so looking at our actions here. And so just going piece by piece, just to break it up while we're moving along here. Please feel free to interrupt at any time. But lu'ua'ea is basically going to mean that we are diving and basically coming back up for air, which we associated with the action of the ROV. And so, and a big acknowledgement to our ROV pilots in the front row as we watched them operate these vehicles live. Secondly, ahiki is basically just a description of like until reaching a certain point. And then the final portion, the papaku. Papaku is a general Hawaiian language term for the sea floor. And although we are kind of, we're exploring sea mounts, as Andy Collins had sort of explained to us in the shift before, yeah, Mauna Kea being the sea mounts, we are generally doing an exploration of that area. And so I believe that the intent was in the naming of these things was to just sort of incorporate more so description of our actions, but as well as the general area that we were working in order to acknowledge those places. So, mahalo.
Mahalo.
Yeah, here's a nice big pillow feature.
Not seeing a lot of coral in this region. Could that be because there's not as much current? There's too much current?
I doubt there's too much current. Yeah, it could be due to a variety of factors.
Yeah.
Looks like there's a couple though.
There's a few.
Yeah.
Something white up ahead.
It looks like in high pack there's a little kind of pinnacle feature we might come up on at some point.
Where's that?
Might require another ship's move to get to that.
Yeah, where are you looking to?
To the southeast of where I think the targets are for Herc. I'm like looking over your shoulder, so I'm not sure that I'm
Oh,
seeing.
okay. Yeah, I don't know if you have one over.
Oh, yeah, right.
It's over on the left side, too. You can check it there.
Yeah.
Yeah, so we have Argus and Hercules are down here.
Yeah.
And then we're kind of heading south right now and then going up this ridge. But
exactly.
Please, if you have any other requests, we can certainly.
Yeah, no, we're definitely moving in the right direction. And I think Herc and Argus are heading right where I was thinking we would.
Awesome. And as always, if y'all in the back row see anything that you'd like to zoom in on as we transit, we certainly have time for that.
Okay.
Steve, I know you're in the data logger seat. Is there anything you're particularly excited and hopeful of seeing?
You know, we've seen a few things that have surprised me so far on this dive. We saw a few different families and genera that, were a bit deeper than I would expect to find in this area, but, it's still pretty characteristic of, these rocky seamount habitats.
Video, could we zoom in on that feature just to the right of where the lasers were?
Sorry, which one?
Sorry, yeah, let me get my this thing.
The coral?
Yeah.
Yeah. Well,
Seems to be another one
just
above it too.
get set up here.
Go ahead and push on in there a bit there, Jeff.
Figure we could look at that while Steve was talking about animals.
Yeah. Perfect.
Try to zoom in and see what that is.
Stand by.
Yeah, so we did make a collection earlier that was really surprising to me, namely this anemone that was on this rock. We collected on our second rock we picked up, and it just looked really bizarre. It was it had some characteristics of tube anemones, but
Come a little wider, please, Jeff.
it was not
Forward.
sediment dwelling, and it was not a ceryanthid at all. Ceryanthids are a group of anemones that have been known and been observed to dwell in sediment, but they have a very distinct polyp or
Jeff again.
tentacle structure. They have 2 rows of tentacles, but this one didn't have that. So it was really a kind of a really unusual observation. So we're hoping that we can find some more information about this when we get back up on the ship. And
The base had 2 colors too. Is that unusual or?
it's probably, part of it was just thicker tissue. It looks more opaque in cross section.
Really moving around a lot, the corals are. So
Yeah.
you can see what Jess is battling.
All right, full wide, please.
That coral colony is one we've seen before on this particular dive. It belongs to the genus Romulogorgia, which was recently created to accommodate a few different species previously belonging to this genus called Pleurogorgia. And since Pleurogorgia has been elevated to a family level, a different family, and that species Romulogorgia has been placed within the Chrysogorgiids, which are the golden corals, which are what these other bottle brushes that we're seeing on the seafloor
More orange-like one. That one in the, talking about that fluffy thing?
Oh, sorry.
No, I was just listening to Steve's description there. Yeah, is that what you were talking about, Steve?
Yeah, those are all part of the same family. Yeah, that white fan and that bottle brush.
Yeah.
Yep. We think we did make a positive ID, potentially even a
Go ahead and
species
push in there,
ID
Jeff, please.
on some of these bottle brushes belonging to the genus Chrysogorgia, and the species is Geniculata. They have very distinctive bottle brush morphologies, and if you actually look closely, this one seems to be have some associates along with it, namely these squat lobsters. Usually you find 2 or 3 living within the colony, and sometimes you can even find things like egg cases or within these coral colonies as well. But it requires a very close,
Full wide, please.
steady zoom. But you can start to make out silhouettes from a distance away as well.
Very cool.
Are we okay to keep moving along for a ship move? Just going to keep
Yes.
checking in. Sure. Bridge nav.
Steve, do these look like dead features to you?
Yeah, we saw
push on
some
in, please.
pretty phenomenal ones a bit earlier in the dive, but these are exactly similar. These are large dead sponges that just have fallen down and gotten caught at this, on this little plateau crevice in the rocks.
That's the same bearing, correct? Ryan?
Sponges, like this are
Yep.
typically grow upright and off the surface of the rock, and when they die or when the colony falls, they end up flat on the seafloor.
Full wide, please.
We also saw some younger live versions as well.
Mhm.
We were told that these are glass sponge.
Yeah, probably in the family Fariidae. We often use morphology as a proxy for trying to designate families or genera, but of course it's impossible to know a lot of sponges to species until we get a piece of the spicules and examine it, or in some cases examine the DNA sequences of the sponge.
The sponge, the corals, these are all filter feeders, but we are at 2,544 meters beneath the surface. Steve, what are they eating?
Yeah, that's a great question. If you look really closely, you can see a lot of particulate matter coming down, like raining like snow through the water column. And that's exactly what they're feeding on. It's called marine snow. It's basically large clumps of particles of dead and decaying material that have been drifting down and flocculating together as they drift down from the surface. These large clumps of organic material, and a lot of the sponges and corals, they're feeding in the flows. In the case of sponges, they're able to create some of their own current, but a lot of the larger corals, for example, are using their tentacles to capture a lot of these marine snow particles out of the water column and using that as food.
Thank you so much. Jeff, can you describe what we're seeing in camera 3?
We're currently keeping an eye on what's supposed to be an eclipse.
Thanks.
But
Any luck out there, Jeff?
But no, it's just a white dot right now.
Oh. Kalamehu, we have a question for you over here.
It's a very nice steep feature here.
Yeah.
Definitely tell that there's good current here because there's essentially no sediment on these rocks. They're swept clean.
Yeah, and from an oceanographic perspective, this is probably not what, for some of our longtime viewers who might have watched our work on the West Coast, it was not uncommon to see large storms of marine snow. But here, where the oceans are a little bit more nutrient poor on the surface, you tend to have very clear water and very little of this marine snow, which is why we have such great pictures of Hercules all the way from Argus. Oftentimes, if we were in a higher productivity area, just the amount of stuff in the water column has a tendency to attenuate light.
Can you look at that, please?
Yep.
At the enemy?
Yeah.
Sure thing.
If there's time, I know we're moving
yeah,
around.
No. All good.
Go ahead, please.
Aloha. Sorry. And so there was a question that came in from Bobby from Burbank, and he was asking, so due to the description of the vocabulary words for the eclipse of the Mahina being like or the Mahina being the moon, wholly being darkened.
It's okay.
Just to answer your question, Bobby. And so currently, because traditionally in the language we do not have a lot of these instruments were not present, so there are going to be two general linguistic approaches to these things. The first is going to be what native speakers of previous generations would do, and that would be to use descriptive words to describe basically the action or any sort of or the general description that might help us to identify that. And then, so for instance, it's like we could say ho'olele ike kani, right, which basically means to send down a sound, yeah. And then otherwise, there is also currently what is called the Lexicon Committee, and this is a committee of Hawaiian language experts from various universities and different communities.
Go ahead and put a shot on that coral, please, Jeff.
And it's their responsibility to sort of designate, and pretty much create new words. And so that was also part of the responsibilities of the cultural working group, was to actually translate various portions of this expedition, everything from the actions all the way up into some of the technology that we're using as well. And so as we're listening out there, please bear with us because there's going to be a lot of variations on specific vocabulary words. And so we would also offer that any of our Hawaiian language speakers out there, please send in your comments, send in any sort of feedback that you might have, and other your suggested ideas in the meantime. Mahalo for your question, Mabi.
Thank you very much for that background and explanation.
No problem. Mahalo ya oko.
So for viewers at home, this seamount area that we chose to dive on, we targeted a zone of depth on this seamount to try to understand the distribution of these corals and sponges that we might see. To understand, some of the species have been detected at shallower depths, and so there's an interest in understanding how deep some of these species go. And so that guided our selection of depth that we're aiming for. So I believe we started around 2,700 meters or so at water depth, and currently we're at 2,534. So we're making our way up the side of the flank of the northwestern flank of this seamount. And again, this seamount was mapped in high resolution for the first time earlier today. And so this is the first footage from this part of the seafloor showing us what the seamount looks like and the type of animals that are living in this environment.
See another sponge there on the left.
Yeah, another.
Yeah, Chris was saying earlier that, you know, these sponges, when you see them live, they're quite impressive, but it takes time for these types of things to degrade. So what we're looking at could be, you know, things that have been there for decades that have fallen down and just they take a long time to decay and degrade.
Not to mention that the growth rate to even get that big in the first place would have potentially taken centuries. Is that right?
Right, yeah, yes. Certainly decades to centuries is a pretty good range.
As someone who studies the microbial life in these systems, I find it fascinating that it takes so long for the degradation to happen of this organic material. It makes me wonder if there's a remnant biowarfare, right? Because some of these species make chemicals to prevent predation and other types of biological attack. And so those chemicals may still be remnant and making this food unpalatable.
Yeah.
Interesting. Would there be any interest in sampling any of the dead coral for that purpose?
No, I don't think so. Well, at least not.
Sorry, the dead sponge.
Yeah, not
actively as part of our.
Not actively as part of the research for this expedition.
Yeah. Yeah, our objectives are really about characterizing the life here, whether that is microbial, invertebrate, fish. We have not seen many fish. We did see a cucumber earlier.
I was going to go 155 this next time, so we start to follow up slope here.
Roger.
155.
As well
Bridge,
as
nav.
yeah, and then of course our other objective is really collecting those rock samples for geological and geochemical analysis. Which is a fancy way of saying how old are these rocks, which may give us an age of the seamount itself, as well as what is living on it and what has changed over time, what's happened, what's the story of the seamount.
Ooh, there's a little sea star at the bottom.
Would you like me to match my heading? With the
Yeah, that'd be great. Yeah. We can meander away over there if we'd like.
Yeah, really cool. It's you know, we only have so much resolution on the mapping that we just did, and it's actually it's incredible. But when you're down here, you see a lot of the bulbous features and things that we can't resolve on the mapping. So it's really cool to see this terrain.